Hello, I'd like to welcome everybody to session 6 of the H3Africa meeting. Good evening, good afternoon, good morning to wherever you are. We're going to do 4 presentations, and then we'll do a Q&A after the first 4 presentations. And so without any further ado, I would like to introduce Seydou Doumbia from Mali. And Seydou, the floor is yours.
Yes, alive. Can you see my screen? Let me share screen. Okay, can you see now?
No.
Okay. Okay. You see now?
You're good. We can see you. Go ahead. Please proceed.
Okay. Thank you. It's a great pleasure for me to be in this presentation and presenting the bioinformatics research training and capacity building in Mali over the last 5 years. Actually, the bioinformatics research training capacity building has started earlier in 2000 by a couple of Malian being trained in biostatistics and bioinformatics at Chile. And when returning back to Mali to set up a training program, the first training program which was set up with support from WHO between 2004 and 2007, the African Center for Training and Research on Functional Genomics, which was also supported by NIH and has trained more than 50 junior scientists. One of the famous of them is Fatumo. So between 2014 and 2015, we also set up the African Center of Excellence in Bioinformatics Research with the support from NID. So Mali got the first African center, and which also was accompanied by a graduate training program in bioinformatics. In 2017, so we got support from NIH to support this training program. So I would like to outlining this program. So the goal of our training program is to build a sustainable bioinformatics research training program and to foster the genomic approach to address global health issues. So specifically, we want to enhance the faculty and the researcher capacity in terms of bioinformatics, genomics, and data science, and also to develop a curriculum which responds to the needs of research and training. So we have developed some master degree curriculum training in bioinformatics and based on bioinformatics core competencies developed through this H3Africa. And we also support student master degree and PhD. So our approach has been based on collaboration, as I said, because we start from scratch. So we needed support from faculty. So we got this support from NIH, from H3 network, and from our other partners such as Tulane University. We got also training education, as I said earlier, curriculum development, degree training, with the master and now PhD training. So the course was blended. Initially, it was mostly distance learning, as we were relying on many of our collaborators to support the training. And we have also organized regularly workshops to address the continuous training needs of the researcher and the faculty through also short-term courses, outreach activities and community engagement to recruit new participants, and symposium also to share experience and evaluation with the career tracking of our students. So that's basically a general overview of the faculty. As you can see, we have about 30 faculty, and among this faculty, most of the faculty are from Mali. So we have more than 15 faculty from Mali, and they all has been also through the training of the trainers through the collaboration I described. And our other partners are from US, from Europe, and also mainly from H3Africa network, as you can see in blue. So the trainees, the goal of the training is to produce an enhanced career master, doctoral and postdoctoral training capable of performing multidisciplinary research involving biomedicine, bioinformatics, and data science. So far we have 33 master students and PhD trained in bioinformatics in this program. We have 2 cohorts of 5 young investigators trained through abroad at Tulane. So the goal is also to make some exchange with Tulane. And you can see that the first cohort was 7, and starting in 2015, and now we have 11 actual students in their first year. For the trainees, you can also see now we have 7 PhD students working on different aspects of bioinformatics, from metagenomics to structural characterization of malaria therapeutic targets, to design of new drug, genomic analysis, and also drug resistance on malaria. So the publication of the trainees, we have now our trainee who graduated. We got 3 publications based on malaria, and also some more training publication are coming on manuscripts on different aspects of infectious disease and non-communicable disease. So we also provide seed grants to the student. Right now we have we have provided 2 seed grants to support research for career development. So we have also students who get some award, travel award, and to participate to the conference. So we organize regularly workshop. So that's some workshop, and you can see also the contents of some of the workshop we organize is to provide training in essential and cutting-edge approach used in bioinformatics and data science. So we have organized so far 3 workshops. Symposium we organize also regularly to provide a platform for investigators across West African region and beyond, to disseminate research results, and also a broad coverage of scientific area from genetic to database and genomic disease surveillance. So in summary, our program has now been very well established. The bioinformatics and data science training capacity has been developed incrementally now established in Mali. The student, the curriculum, and the faculty targets provide a platform for new bioinformatics technician, faculty, teacher, researcher, and related personnel. So we have increased the bioinformatics and data science capacity training in Mali over the past 5 years. So thank you for your attention. And one of our fellow will present after me. And that's one of the symposium we organize, as you can see, which attracts a lot of researcher and student from Mali and across the region. So I would like to thank all our collaborators from USTB, from Covenant University, Tulane University, and NIH, and all these people who has contributed to these training grants and the bioinformatics training program in Mali. Thank you for your attention.
Thank you very much, Seydou. That's a great overview of the program. And now we're going to hear from Abdoulaye Diawara, who is a student in the PhD program that we just heard about. So we're ready for his presentation.
Hello, everyone. Myself, Abdoulaye Diawara from African Center of Excellence in Bioinformatics of Bamako from Mali, and I'm going to present on a SNP barcode to inform genetic variation and evolution of the malaria parasite Plasmodium falciparum in Mali. To get started, let's look at the background. Despite enormous effort to combat malaria, the disease still kills countless children worldwide, mainly in the Sub-Saharan African countries. A significant concern is that Mali is among the 10 countries with the highest number of malaria cases and deaths, and the P. falciparum is the deadliest among the Plasmodium species. Fundamentally, the genetic diversity of the P. falciparum population is a major factor in the parasite's ability to adapt to changes in its environment. To explore Plasmodium falciparum population dynamic in Mali, we set to ourselves the following objective. The main was to investigate diversity and evolutionary relationship of the Plasmodium falciparum population in Koina, Bambara, and Nioro du Sahel. And then the specific objectives were to determine the mutation direction in SNP Plasmodium falciparum genome, and to estimate the ratio transversion, and determine minor allele frequencies of Plasmodium falciparum isolate from malaria patients, and assess the genetic similarity between the two parasite populations. Samples used in this study were obtained from two sites: Koina Bambara in the northeast of Cebu City, where malaria is hyper-endemic with a seasonal transition. Nioro du Sahel, in the other hand, is located in the Sahelian region of Mali. Malaria transition is unstable. A set of 101 SNP were examined for map in 3 populations on PlasmoDB database, and then SNP were assessed for the independence from one another. For sampling, we use rapid diagnostic tests for malaria diagnosis, and DNA extraction was done by Qiagen kit, and genotype was determined by using Agena Max. Sample bar coding was done by concatenated genotypes at 27 SNP with 80 isolate. This slide shows the statistic and bioinformatic analysis I have done during this study. And as you can see, the alignments was done on cluster X2, and mutation direction and diversity was done by using Mega X, and the pieces was done by using past software. The phylogenetic tree was generated on the phylogenetic.fr online software. This table shows you the 27 SNP barcode of the 57 Plasmodium falciparum isolate in both sites, after excluding sample with the barcode containing more than 4 missing SNPs. Here I'm showing the evolution of the single nucleotide polymorphism. If you look at this, you will see a higher frequency of interchanges guanine, adenine, and cytosine thymine, and then a less frequency of exchange guanine, adenine, cytosine, and thymine, guanine, and then cytosine, guanine, and guanine, cytosine. This clearly shows that the ratio transition transversion were high at both sides, but particularly in neurodecidal, and indicating that Plasmodium falciparum genes were under the high selection pressure in this population. Here, the diagram is comparing the minor allele frequency. You can see in the both population, all SNP had MAF value higher than 0.1, indicating that this population are highly diverse and show a interpopulation diversity law according to the MAF value of the two populations compared to one another. When the PCS was performed on each parasite population, the point shows more divergence of the parasite. But when all samples were considered, the parasite appear to be grouped around the same region, indicating similarity between the two populations. Phylogenetic tree analysis showed that samples collected from both sites come together into 5 major clades. This lower genetic divergence observed could be due to similarly selective pressure from the various control and management measures within the country at large. In conclusion, genetic diversity facilitates parasite populations in environmental adaptation. Results here may partially explain the rising drug resistance and the increasing severe cases in Koina-Bambara and neurodecidal regions in Mali. I would like to express my special thanks of gratitude first to NIH Forgat Grant and Professor Seydou Doumbia, Professor Mahamadou Jakte for founding this study. My profound gratitude goes also to Professor Mamadou Wole, Dr. Talib Youssef for their help. And I cannot end this presentation without giving a special thanks to Shefre Jeffrey from Tulane University. Thank you for your attention.
We're going to hold the questions until we've had 2 more presentations. The next presenter is Fred Sarfo from the West African College of Physicians, where he had his degree, is going to present on novel insights into determinants of stroke risk and outcomes in Africa. He works with PI Mayowa Awolabi.
Hello, and welcome to this presentation. In the next 8 minutes, I'm going to share with you some of the novel insights into the determinants of stroke risk and outcomes in Africa on behalf of the SIRENE and the SIPS genomic studies. This work has been going on for the past 8 years, and I want to share with you some of an overview of the key findings so far. Well, SIRENE and SIP Genomics have, for the past 8 years, been focusing on trying to understand what are the determinants of stroke risk in Africa and what are the genomic contributions to stroke occurrence on this continent. In this presentation, I'll be focusing mainly on the risk factors and the outcomes of stroke. And by way of acknowledgement, this work was done in Ghana and Nigeria across 15 hospitals, a lot of community engagements, a lot of investigators have been involved, and we are eternally grateful to them. So by way of background, Africa, as we now know it, has the heaviest burden of stroke on the globe. The age standardized incidence of stroke is 316 per 100,000 population. The prevalence rate of stroke is 1.4 per 1,000 population. Our case fatality rate is 30% within a month of stroke onset. And one study from Tanzania shows that the stroke fatality within 3 years is 84%. So the outcomes and the prognosis for stroke are really grim on the African continent. So when we started out in SIRENE, we wanted to look at what is increasing the predisposition to stroke occurrence. Of course, we know that the risk factors such as hypertension and diabetes were contributing to stroke occurrence, but we didn't know the population attributable risk for these risk factors or the odds ratio for these factors. So one of the key findings from SIRENE has been to quantify the effect sizes of 11 dominant stroke risk factors in Africa. And these include hypertension, dyslipidemia, meat consumption, obesity, diabetes, low consumption of green leafy vegetables, stress, table added salts, cardiac disease, physical inactivity, and then cigarette smoking. In the next slide, I want to share with you the effect sizes of these dominant risk factors. This work was published in the Lancet. And when we look at hypertension, for instance, the odds ratio is 19.36. And the population attributable fraction for hypertension is 90.8. So hypertension is way, and it is topping the charts. It's just breaking the charts. So 90.8% of population attributable risk for stroke occurrence in Ghana and Nigeria, at least, is explained by hypertension. And then we have dyslipidemia with a population attributable risk of about 35.8. We have diabetes, population attributable risk of 22.16. Obesity, population attributable risk of 26.5%. Vegetables, green leafy vegetable consumption that is not sufficient, contribute to stroke, and the population attributable risk is 18.2%. And then regular meat consumption, which are dietary factors, also contributes about 32% to the occurrence of stroke in Africa. So when we look at specific stroke types, which is ischemic stroke and then hemorrhagic stroke, what we found was that 10 of the 11 risk factors that were identified in SIRENE were associated with ischemic stroke, and then 6 out of the 11 risk factors identified overall for stroke was also associated with intracerebral hemorrhage. And here on this slide, I show you the population attributable risk of these risk factors for ischemic stroke on my left, and then the intracerebral hemorrhages on my right. And there again, we see hypertension topping the charts. and then significantly inappropriate or low consumption of vegetables also contributing significantly to the occurrence of stroke in sub-Saharan Africa. We're also interested in the risk factors for stroke occurrence by country. As you know, this was a two-country study. And we see here that if you look at Ghana and Nigeria, hypertension, diabetes, lipids, which is dyslipidemia, and low vegetable consumption was co-shared between the two countries. And then if you look at Nigeria specifically, obesity, stress, meat consumption, and regular sugar consumption was associated with stroke in Nigeria, whilst in Ghana, cardiac disease, physical inactivity, cigarette use, and salt consumption was associated with stroke in Ghana. So what are the stroke types that we saw from Siren? Well, if you look at the West, typically about 90% of strokes are ischemic and 10% are intracerebral hemorrhages. In Ghana and Nigeria, 70% of our strokes are ischemic, 30% of them are hemorrhagic. Then if you look at the etiology subtypes, if you come to or focus on the ischemic strokes, we see that small vessel occlusive disease or lacunar strokes are by far the commonest stroke subtype in ischemic stroke, followed by large artery atherosclerotic disease, followed by cardiac disease in purple. About 30% of the stroke subtype in ischemic stroke were undetermined. Now, to my far right, if you look at intracerebral hemorrhage, by far, hypertension contributes about 80% of all hemorrhagic strokes. So the rest, such as aneurysms, cerebral amyloid angiopathy, medication-related, and undetermined, contributes less than 20% of all intracerebral hemorrhage. So the message is hypertension is driving almost all the intracerebral hemorrhages that we see in Africa. If you compare the proportions of ischemic strokes that are due to small vessel occlusive disease, which is the commonest in our study, it is comparable to data from Pakistan, from China, and also from Japan. As you move to the more industrialized country, you tend to see more cardioembolic related ischemic strokes, such as in Germany and then in the UK. So with all this information on stroke risk factors, we're able to develop what we call an Afrocentric stroke risk prediction. And here we use 15 of the putative risk factors, and we're able to calculate a Cohen's kappa score for reliability at a maximum stroke risk of 56%. So with our riskometer, it had a predictive accuracy of about 76%, sensitivity of 80.3%, specificity of 63% in predicting stroke risk using risk factors that an individual person may have. And then if we focus now on stroke outcomes and determinants, out of the over 3,700 stroke patients that were enrolled into Siren, we're able to identify that 21.8%, nearly 22% of them died whilst in hospital. And the 5 factors which were associated with stroke fatality were low vegetable consumption. So this again highlights the potent contribution of vegetable consumption to stroke occurrence and even to outcomes. And the other factors which were not surprising were stroke severity. The NIH-SS score for each 5 units increase was associated with an adjusted odds ratio of 1.63. When we used cerebral decompression therapy, such as mannitol, it increased or associated with stroke risk mortality of 1.98, elevated intracranial pressure 3.71, and then aspiration pneumonia. We are in the process of writing up a manuscript that is looking at the system-level determinants of stroke risk, and we'll be sharing that with you in due course. So what are our future perspectives? Well, looking at all the data we now have, it is now time for us to develop and test interventions for primary and secondary prevention of stroke on the continent. And then we also need to develop long-term stroke cohorts to be able to understand the determinants of stroke outcomes in Africa. As I mentioned, the GWAS studies that we did in Cyrene and it's ongoing in SIP Genomics, we are almost a few weeks away from releasing the first manuscript of the novel genomic markers for stroke. So watch out for that. And I want to thank all the organizers, the NIH for funding. I want to thank all the collaborators for this study. Thank you very much.
Thank you very much for your presentation. We are scheduled to have a live presentation from Dr. Akin Kunmi Okakunli, but we don't seem to have Dr. Kunli online at this point. So I think what we will do is move to the Q&A session. And I see we have our three presenters here to answer questions. So let's start with the first question, which is for Seydou. It's a very nice compliment, actually. It says, this is from Sigun Fatumo, who said, "Great presentation, Seydou. 16 years ago, in 2005, I had the opportunity to learn bioinformatics in your workshop in Bamako, Mali. That was a great experience. Since then, I have become a faculty in your Center of Excellence. Thank you for providing a platform for career growth." Now to my question, but that's a very nice compliment. Congratulations, Seydou. The question is, what are your strategies to retain individuals that are trained in Mali to remain in and contribute to your institution and the country?
I think it's student like alumni like you encourage anyone to pursue supporting the training. This is a very challenging question, particularly when it comes to bioinformatics, a very growing field, but still lagging in Africa, and particularly when you train people and there is less opportunity beside the research now, which is also limited. So that's why we rely more on the selection process. So we try as much as we can to recruit our pool of students from different research programs. So therefore, that's facilitate their integration. So, and also it helps identifying research questions which related to what they are doing. So that's one strategy we are also doing. Another strategy is also as we provide some seed grants which help them to build a career. And as I said, also we do more outreach so that people be aware of what bioinformatics can do, particularly in this genomic arena. So to create demand, and what we also do for the student is also to encourage them through the networking, whether it's Mali, but also in Africa, that they can be, because if we build the program for only Mali, so at some point the job market will be full. So then we also encourage students through the different network and collaboration, so that give them opportunity for job. But it's I do recognize that it's a very challenging, but so far we have been quite successful by tightening them to existing research program or teaching.
Okay, thank you very much. This question is for Abdoulaye from Michelle Skelton. Says, when you say more severe cases of malaria, can you provide more detail of severity as opposed to the less diverse parasite?
Okay, thank you for this question. I hope you can hear me.
We can hear you.
Okay, thank you for this question. Really, I didn't check more on that, but in our understanding, we say to ourselves, if malaria is not controlled, but can lead to several cases in the country. That is why we think that the diversity can be explained a part of this situation in our country. But we are going to check with other study to see if several infections are more commonly multi-clonal across the country.
All right, thank you very much. This
Yes,
next question
please.
is,
Perhaps I can add on that in general, where we have less diversity, it's also where we have more severe cases. Where we have more polyclonal infection, that's disease is more endemic there, so it's less severe. That's what we see in general in the field. To add
right,
to our comments. Thank you.
Okay, thank you, Professor.
thank you. The next question is for Fred Sarfo. It comes from Michelle Ramsey. It says, you mentioned that stroke prevention intervention is one of your next objectives. Since hypertension is the largest contributor, what would your strategy be to address that?
All right. Please, can you hear me?
Yes, we can.
Yes, thank you very much. At the heart of the issue with hypertension in Africa is lack of awareness. A lot of our folks are not aware they have hypertension, so we see very young patients, 40 years, 50 years, come down with hypertension. So the focus now is on how we can increase knowledge or improve knowledge based on individuals that look, hypertension is asymptomatic. So we need to do regular checkups. And these checkups can be done in places like churches, mosques, where people go every week for service. This is one area where I strongly believe that we can target people and identify those whose blood pressures are high, so that we can link them to nearby clinics for follow-up. Having said that, in addition to increasing the level of detection of hypertension in the community, we also need to look at how even patients who are attending clinics would adhere to treatment and be able to get their blood pressures controlled. We've done a lot of studies in Ghana that shows that even those in clinics, just about 50% of them have their BPs controlled. And a lot of it is from therapeutic inertia. Clinicians don't increase their BP medications when it's not clearly achieving the target. And number two, patients are not adhering to treatment. Some of the things we've been doing, and one of the studies we are currently doing, is to see if a nurse-led guidance can improve, nurse-led education and guidance can improve BP controls in a community where patients can check their BPs and be able to report back to the nurse that, look, the BP I checked is still high, so what should I do? And then the nurse can then coordinate the level of care. So enhanced detection of hypertension in the community, improved control in the clinics where patients are being treated for hypertension. I believe those are the key ones. And of course, the lifestyle risk factors. We know that salt intake, as we show in Siren, is strongly linked with poor outcomes of stroke and also linked with stroke occurrence. So if we can cut down salt intake, take more vegetables. Okekule will be talking about dietary predictors of stroke in Africa. Those, I think, we can implement through mass media, educating the public the need to cut down salt, exercise more, increase vegetable intake. And those, I believe, may be able to help. Thank you.
We actually have another question for you, Fred. The question is from Bamadile Tayo. The question is, are the reported odds ratio for hypertension in your figures adjusted for other risk factors?
Yes, please. So these are adjusted odds ratios. So all the odds ratios presented are adjusted for all the 11 other factors that were associated with stroke. Which is one of the things we found quite striking, that the odds ratio was about 19. One of the reasons potentially contributing to this is that we measure blood pressures when patients were admitted, and we didn't have readmission blood pressure because of the design of our study. It was a case control study. Having said that, there are ways you can adjust for blood pressures in stroke patients using the Fisher's coefficient to be able to get a sense of what the baseline was like before the stroke. But it's still quite high, and it's still quite, we think it's probably one of the highest in liquid.
All right, thank you very much. So our fourth speaker in this session has been able to log on. And so I'd like to now introduce Dr. Akin Kunmi Okekunle. And the floor is yours for presentation.
Yes, I'd like to thank everybody for making me to join. And my apologies for joining late because of logistics. So, good day, everyone. I'm Akin Okekunle from the University of Ibadan. I'm making a presentation on unraveling the risk factors of unraveling the effect of diet and cardiovascular. So, the title of my presentation is Unraveling the Effect of Diet on Stroke and Cardiovascular Disease in Africa, which is primarily on the basis of our findings from the Stroke Investigative Research and Education Network, where Professor Mayowa Wulabi is the principal investigator. First, I'd like to say that we do not have any conflict of interest with respect to this presentation, which I'll be making under the following outlines. By way of introduction, stroke and cardiovascular disease is one of the major risk factors for mortality worldwide, and several factors have been implicated in the burden of cardiovascular disease and stroke. However, little effort has been made towards identifying the role of diet in the epidemiology of stroke in Africa, and to that effect, the main goal of this presentation is to present our efforts on the role of diet in stroke and cardiovascular disease among indigenous Africans. And these presentations are derived from some of our published efforts, which you may want to consider in the near future to look at after this presentation, and some of our unpublished results as well. Earlier, we had already found out in our match case control design that frequency of vegetable consumption was associated with a lower risk of stroke. And on that note, to test this association further, we attempted a meta-analysis that was prospectively registered to test the association between higher intakes of vegetables and risk of stroke and cardiovascular disease. And in addition to that, we attempted a match case control design. 3,000 stroke cases were matched with stroke-free controls from Nigeria and Ghana, taking into account. And in that population, we attempted the principal component analysis to where we identified 7 dietary patterns, and we attempted regression to test between these dietary patterns and stroke, adjusting for relevant covariates in the study. So our first finding in our meta-analysis revealed that higher consumption of vegetables were inversely related with stroke risk independent of a type of cardiovascular outcomes, sex, and quality of studies. Although case control studies had poor performances, but by and large, the core studies provided reliable inverse relationship between higher vegetable consumption and risk of stroke. Case control design from Nigeria, where we recruited respondents from Nigeria and Ghana, we found out that stroke cases presented higher prevalence of smoking, physical inactivity, and alcohol use compared to controls. And moving to the result of our conditional logistic regressions, we found out that heavily meat-based diet, red meats, poultry, and organ meats-based diet were associated with a higher risk of stroke, while fruit-dense diets, whole grain diets, and vegetable-dense diets were associated with lower risk of stroke. By and large, you will realize that vegetable presented better performance in terms of statistics in terms of odds ratios in this relationship. And moving forward, we attempted to 7 dietary patterns in a single model, and we adjusted for relevant covariates such as hypertension, diabetes, and the likes. And we found out that 2 dietary patterns stood out distinctly. First was the fact that poultry product, meat-based diet as well was associated with about 30% stroke occurrence, or about 1.3 odds of risk of stroke, while vegetable dense diets were associated with a 40% less risk of stroke in this population. And if we were to summarize our findings, primarily we find out that meat-based diets and fried food and sweetened drinks were associated with higher risk of stroke and cardiovascular disease, while vegetable dense diet, fruit dense diet, whole grain diets were inversely associated with the risk of stroke. And in all of these associations, vegetable dense diet stood out significantly. And one key finding I've established here is the fact that it is likely that regular consumption of vegetables might be beneficial as a primary prevention strategy in lowering the risk of stroke and managing the burden of cardiovascular diseases in our population. And future consideration for our study will be to test these associations in multi-ethnic cohorts of indigenous Africans, and testing the significance of diet as epigenetic markers of stroke occurrence. And on that note, I'd like to acknowledge our key investigators, where Professor Mayowo Labi stands as a PI. You have had Professor Fred Sapo's presentation earlier, and our PIs, our investigators are from different institutions across Africa and in the United States. I'd like to also acknowledge our funders, the National Institute of Health, the H3Africa, and finally to also thank you for paying attention to this presentation. Thank you very much.
We'll hold questions until after the last presentation. So I'd like to now introduce Rolanda Julius from the H3Africa Coordinating Center for an H3 Africa Coordinating Center update.
Good day, my name is Rolanda Julius, and today I will present a snippet of the H3Africa Coordinating Center Monitoring and Evaluation Plan. So one of the major goals of the H3Africa Coordinating Center is to monitor the project outputs, to evaluate the progress made, and to report on the impact of the consortium at large. Over the years, we have collected a number of aggregate data, so we have an idea of our membership growth. We have the number of trainees. We have the number of trainees supported through travel fellowships over the years, and we have the publication output as well as participant recruitment. Now we would like to have a more granular view, and this will better be able to demonstrate the impact of the consortium. So our objectives are to capture the extent of capacity building, both in human resources and infrastructure. We would like to capture the successes of the project, and we would like to identify the areas for improvement or in need of intervention. H3Africa currently has 52 projects. However, not all of the projects are currently funded. There are 4 broad categories of projects: our biorepositories, our LC projects, our training projects, and our research projects. The research projects are further categorized into individual research projects as well as collaborative research networks. And we would hope to capture information also of all the sites in the collaborative research networks. We will conduct a survey using computer-assisted personal interviews, and this will take place in our Zoom rooms based on our appointments with our respondents. We will also make use of self-administered web-based surveys, and these will be programmed into the H3DB and then circulated as a link via email. So we will have 6 questionnaire sets, 1 unique per project type. So a questionnaire set for the research projects, 1 for the training projects, 1 for the biorepositories, and 1 for the LC projects. In addition to that, we will have a questionnaire set to capture career development of our trainees, as well as the individual career development of other project members who are not trainees. Each questionnaire set should take no longer than 10 minutes to complete. This is an example of the questionnaire set for a research project. This is what we call the paper-based version. So we would hope to make use of the information already captured in the H3DB to pre-populate the question sets, and this way it will make questionnaire much shorter and less time consuming. So our approach will be to identify our respondents. We will do this by initially looking at the project roles of the members as indicated on their user profiles on the H3DB. Of course, we will confirm with the PIs whether these people identified in this way will be the appropriate people to be able to answer the questions related to their projects. But of course, the PIs will also be able to nominate the people that they would like us to interview to answer questions based on their projects. We would hope that these respondents will also assist us to carry out the web-based surveys for their project members. And these questionnaire sets, of course, are the trainee question set to capture individual career development of trainees, as well as to capture the career development of the other project members who are not trainees. We would also like to identify our enumerators from the consortium members, because we know there are many who are experienced in carrying out surveys. Of course, we will also provide some training and guidelines, and of course, the resources to be able to carry out the survey. So this is just our rough timeline. Questionnaire design has started back in 2018, So currently we are just refining it. We will then identify and invite our respondents in this month, and then we will start to identify and train our enumerators. Then we will start collecting the data and pilot and revise the survey. Then in October we will start the survey, and hopefully by November we will be able to have some preliminary analysis of our results. And then hopefully in early December we will be able to have a first report of our findings. We would hope at the end of the survey that we will be able to provide a summarized output of our findings to each participating project, which they may be able to use for their reporting purposes. We would hope to be able to develop targeted interventions should these be needed. We would hope to have an idea of the collective infrastructural capacity that has been built up over the years under the H3Africa Consortium. And we would hope to have been able to generate material to use to promote the consortium to our potential investors, as well as to communicate to stakeholders and also potentially to stimulate new research interests. Overall, we would also hope that this will emphasize the need for sustainability of the H3Africa Consortium. So our next steps are that we will start making appointments, and we hope that you will set aside some time for these and support us. And we will be tasked to design the questionnaire set. However, we would like you to ensure that we ask the right questions when we pilot the survey with you. We, of course, will provide the platform, and we hope you bring your enthusiasm. And we, of course, would like to train and compensate enumerators, and we would hope that you will sign up and get involved. We also hope that you will provide accurate information, and we would ensure to communicate this fairly. I would like to thank our funders for their continuous support, as well as the projects we know that will support us in this endeavor.
Let me look in the chat here. I think we have a question for Dr. Onakule from Bamadile, Taiwo. Not sure if I missed it in your presentation, but what was the effect of SES? One would expect that some of the risk factors to be correlated with SES.
Okay, actually, because of time, first and foremost, let me thank you for your question. Actually, because of time, I would have made such presentations, but we found out that the socioeconomic status, particularly income, had some correlation at some point. But what we did in our analysis was to stratify by low socioeconomic status and high socioeconomic status. And we found out that that association between vegetable dense diet and risk of stroke was still consistent as in the overall data compared to high income population. And perhaps maybe there are several reasons that could account for that. Primarily, perhaps maybe because a population might likely be from an agrarian setting where they have access to vegetable consumption than high-rich society like urban settings where that the vegetable for access to vegetable may be very, very expensive. So that's something we're actually looking at. And the same thing we did for physical inactivity as well, but that did not have so much, it didn't part up that association. Thank you.
And there was one more question for you. And it was, if diet appears to play such a significant role in stroke outcomes, has the influence of the microbiome been considered in your research efforts?
No. That is what we're considering in the future. Of course, you know that considering microbiome is actually something good to look at how vegetable consumption manipulates gut microbiodata. But we currently don't have funding for that yet, but that is something we are critically looking at in the future. Thank you for that. Thank you.
Okay, I think that covers all of the questions that we have. We'll give a second for anyone else to ask any. All right, if not, I thank all the presenters for their great presentations in this session. And we can call this session concluded. And I understand that there is a Zoom session now where the IEC feedback will happen. So thank you very much to everybody and for all of the participants.
Thank you.